is a postdoctoral fellow at the Wyss Institute at Harvard and also the Allen Discovery Center at Tufts University. Our topic today will be Sim2Life, AI-generated biological constructs. And just as a special note, this work recently won the Cosrelli Prize from PNAS in Engineering and Applied Sciences and was also featured as a museum exhibit in the London Design Museum. And we'll go ahead and get started.
All right. Can everyone see my screen?
Yep, I think we're ready to go.
And me? Okay. So I can only see my screen. Feel free to interrupt Lisa in the beginning if you need me to adjust. There's also a tropical storm outside of Doug and my window. We're in separate locations, but in the same city. So apologies for thunder or dogs barking. Besides my dog, there are two other members of our team that are not present today. Pictured here, Josh Bongard and Michael Levin. And I'm Sam Kriegman, and I'll start this talk talking about the AI side of Sim2Life, and then I'll hand over the floor to Doug Blackiston at halftime, and he'll talk about the biology, the life side of this. So there has been much work in AI-generated systems, such as AI-generated images and movies, and much work in the literature has investigated how to generate interesting art and realistic looking but fake images, and also how to detect whether or not an AI or a human was the one that generated these artifacts. There have also been some work on generating physical systems, such as evolved antennas that can pick up unusual radiation patterns that are difficult for traditional antennas or existing antennas that were human designed to capture. There have also been AI-generated robots, and this is the field of evolutionary robotics. However, for the field's 30-year existence, it has rested on the assumption that those robots, if they're going to be built at all in the real world, are going to be built out of robot stuff, metal, plastics, electronics, chemicals, those kinds of things. So in this work that Doug and I are presenting today, we're going to relax that assumption. And we're going to allow AI to generate biological constructs instead of electromechanical ones. And here is a picture of one of those AI-generated blueprints on the left, which is built out of these two kinds of building blocks that we'll describe in this talk. And on the right, you can see the actual living system that was built by Doug to capture the same overall shape and internal distribution of these cell types, and also its function, which is not pictured here, but you'll see in the videos. So I want to take the first half of this talk, my time that I'll be speaking, to talk about the 10,000-foot view. What are AI-generated biologicals? Where do they sit in the wider literature of synthetic biology and robotics? Why one might want to make them, and how they're designed by AI. Then I'll hand over the floor to Doug, and he'll talk about how they're built and the biological underpinnings of this work and implications. So again, we have our little design here, and those red blocks were our cardiac muscle that expands and contracts, and the ones on top are skin tissue, which is passive. And if put together in the right way by the computer or by a human, it will behave in some way that might be desirable, such as moving into the right-hand side of the screen. This design is sent over to Doug, and Doug builds a prototype that matches the AI-generated blueprint by positioning the real cardiac cells and the real ectoderm cells in the same positions as proposed by AI. And in this case, as you can see in the bottom row here in the video, the living biological construct behaves in the same way as its simulated AI-generated counterpart. To give you a sense of the size scale we're talking about here, these are very small. They're 0.5 millimeter across, and you can see them with the naked eye, but they're very, very tiny. They're examples of synthetic biology, which is a well-established field that continues to prove very useful and interesting, but is dominated by a paradigm of human-designed synthetic biological objects. And those objects are organisms or part of organisms, and biohybrids, which combine synthetic and living matter into a single functional object. The parts of organisms that have been explored are organoids, which are not self-motile. They are kind of recapitulations of organs in a dish, like a liver or a kidney or a lung or cortical tissue. They've been very useful for studying disease and treatments for those diseases, but they can't move on their own through their environment for the most part. And this limits the kinds of applications that they could be used for to produce useful work. There have been biohybrids that are self-motile, and the way that most of them are made is they take a silicone scaffold and they embed rat heart muscle cells into them. So the heart muscle cells in this case were optogenetically transfixed to respond to light. In other words, when blue light is shined on them, the heart cells contract. So if you pulse a light on one side of this stingray, it will move away from the light, reminiscent of one of Bratenberg's vehicles. But this was human designed, and human designers tend to design things that are familiar to them. So it is not surprising that a stingray or a jellyfish or something that is known in the world to be able to swim was selected as a design. And it's also just one design instead of a whole palette or design space of possibilities that could be proposed by an automated system. So I'm going to bring everyone over to the left side of this tree, which is the AI-generated synthetic biology, which could be broken down into organisms and biohybrids. And in our case, what we made were called reconfigurable organisms. That was the technical term in the paper, because these cells, as Doug will explain, were taken from very early frog embryos and reconfigured into new ways. That this could be done in different ways. Instead of reconfiguring cells and treating them like Lego blocks, you could potentially grow new organisms. An AI or human could direct that growth. The particular cells that we're using come from Xenopus laevis, and hence the name in the popular press, xenobots. So why would you want to do this? Well, if they could move around and do something useful in the environment, they could be used as machines, as robots, but made entirely out of biological components, they would be biodegradable. Unlike existing technologies, which are made out of plastics and chemicals and metals, and these kinds of things degrade over time, and they have very harmful ecological side effects. Another area that is intriguing is the possibility of building these biological machines out of a human patient's own cells, so that it could potentially deliver medicine or internally ablate tumors without being rejected by the body's immune response system. But there's also the basic science question here of understanding adaptive behavior and intelligence and figuring out what else is out there. This might not yield any useful applications in the short term, but it will expand our knowledge, and that knowledge will ultimately be embodied in future technologies that we can't even imagine at this point. I think that it's very important to do this basic science because there's so much to learn about adaptive behavior and intelligence and how to build artificial intelligence. If you think about chemistry, it's a well-established field. It has the periodic table of elements. Biology has Darwinian evolution. Physics has quantum mechanics and relativity. What about AI and cognitive science and robotics? It seems like we're missing that touchstone fundamental theory. So hopefully this work, by creating new kinds of biological entities, can help us expand that knowledge base and ultimately arrive at some kind of useful theories and explanations. So concretely, we're going to use two very simple biological elements, skin and heart muscle. And we treat these as building blocks. So you might, or an AI might be presented with a collection of these blocks and asked to build something. Well, if you build some kind of structure out of just the skin tissue, which is made to be passive, then it's just a statue. So it's predictable. We know what it's going to do, but it's not necessarily useful. We could also build known structures or let these cells become what they want to become. So a heart made out of heart cells. It's predictable. We know what it's going to do, but it's limited to a single task. It can't walk across the street and get your mail. But the problem here is to be able to design new kinds of structures with existing cell types. We don't know how the cells are going to coordinate and how they're going to behave when they're interlaced with skin and heart muscle and new shapes and forms and functions. So we're going to model this system. We're going to allow the heart muscle to volumetrically change in the model. And we're going to give our AI a goal of how to arrange these blocks just to move into the right side of the screen. And we can see here that when the AI puts together random configurations of the heart cells and the skin cells, they don't tend to do much. They don't fully satisfy this objective of moving to the right. But you can see that the one in the right-hand side, the right column, is a little bit better at moving. It tilts over just by chance. And so by replacing the worst designs with modified copies of these better ones, we can make incremental progress. One other thing to note here before I show you the progress made by an evolutionary algorithm is that we don't know how these heart cells are going to coordinate their actuation in space and time. So this has been modeled as random. So this is known as dynamics or domain randomization. So we don't know how the real heart muscle cells are going to behave in these kinds of novel configurations. So we just randomize it. And after evolving designs in computer simulations for a couple of days, we get designs that, despite the random movement of the individual heart cells, the overall shape of the organism and distribution of passive and active tissue coheres this random behavior into a organism that can move into the right-hand side of the screen, which was our goal. So I'm going to dive down into the details here very quickly of how those designs were generated. You might have noticed that even the random ones looked kind of coherent. They had structure. They weren't just random blocks. They were bodies. So this is produced by what's known as a compositional pattern producing network. So this, in the simplest case, we can just hook up some kind of spatial coordinate to some output we're interested in. So here the X coordinate is hooked up to this 2D space of X and Y, and outputs how much green is in the pixel. So you get a gradient from no green to very green. You do the same thing with the Y coordinate, white to green. You can add interaction terms in this network, or you can allow the AI to figure out what interaction terms should go in this network, such as a threshold that switches between white and green. If you start composing more interaction functions into these systems, which have input in this coordinate space you're interested in and output something that you want on the other end, you get interesting patterns here, some kind of green snake. You can change the output to red, green, and blue, and get some very beautiful patterns by just changing the structure of the network. Likewise, you can add in a third dimension to create 3D objects. And that's exactly what we did here with our biological constructs. So with the input X, Y, and Z in 3D space, we might have a somewhat more complicated network than the simple ones that I was showing. But at the end, what's going to happen is, instead of painting green into a cube, we're going to say, is there biological stuff at this location? And if so, what kind of stuff? and it places a voxel, which is just a 3D pixel at that location. So it might say at X1, Y1, Z0, yes, there is material there, and it should be passive skin tissue. But next to it, for whatever reason, the network outputs nothing. On top of that block, however, it says there should be contractile tissue. You can query every space, every coordinate in this workspace. In the end, you get a design. That design is then simulated in a physically realistic environment, automatically assigned a performance score. And if it's good, it is saved and sent off to Doug to be built. And because we're using AI, we get many designs that all satisfy the objective, but they're diverse and move in slightly different ways. And so that's something very interesting that you get from this system. And at this point, without further ado, I'll hand over the presentation to Doug so he can tell you about the fun part of building these in the real world.
Hopefully we're in business.
Looks good.
Great. So I am a biologist, and I came at this project from a slightly different angle in that I had seen a lot of these types of constructs and designs that Sam had been building for a number of months or some of our collaborative meetings. And I had this question of, could we build these out of biological material? Could we build a biological robot, which had never been done before? And this was really just a thought exercise at the beginning, but has become the focus of a lot of the work that we do in the lab now. So the big question was, how can you even build an AI-generated biological construct? As Sam has says, there's existing systems that do this a little bit, these biohybrids, and there's generally two parts, these synthetic scaffolds, which form the morphology. So in the swimmer down below, this is from the SAFE lab at the University of Illinois. The backbone of this swimmer is made from some sort of artificial component. And then biological actuators are layered on top of these scaffolds. So in this case, cardiac muscle. And the generation of force from those muscles allows this micro swimmer to move through an aqueous environment. That really wasn't going to work for the types of designs that had come up out of the simulator, because these are completely soft-bodied robots of all different shapes and sizes. We can't standardize the synthetic component. So the goal became to create a method to design a fully biological robot composed entirely of cells with no artificial components. And so this really is using developmental techniques to create an entirely synthetic organism. And as Sam alluded to, there's two big advantages here that we get as a side effect of this design principle. One is that such a creation will be biodegradable, so there's no artificial components after the end of lifespan that are left in the environment. And also these could be biocompatible. So in a patient setting, these could be made out of human stem cells or any animal cells that are needed. And you could use these, and these could either participate in the development of the organism when they're finished, or simply break down harmlessly and not be attacked by the host immune system. So this is easier said than done. How do you do this? We came up with a novel approach, and the idea I had was, could we simply deconstruct an organism into component pieces and then reconfigure the components to build a novel structure, a new living organism? And so throughout all of my work, my component source has been the embryos of the African claw-toed frog, Xenopus laevis. This is an aquatic species. I'll show you a video of this process in a moment. But if you take a developing frog embryo, our frogs live about 20 years in the lab, and they produce 3,000 to 4,000 eggs. So there's a rich source of external stem cell material that we have access to. And after about 24 hours in this cartoon, what you would see is over here, this pigmented area is the top, and this would be the bottom if you were looking at it from the side. You get a few thousand stem cells. And these look relatively the same, but from a number of developmental biology studies over the past 100 years, we know that there's actually already an atlas or a fate map that you can layer on top of this. So these cells are pluripotent. They can become any tissue type. But if left on their own and not given any molecular intervention, these different regions will become different tissues. So the top will become skin and nervous tissue, the middle generally becomes the muscle, and the bottom becomes the internal organs. And so you could imagine taking a developing embryo at this stage, breaking it apart into individual green, purple, and red cells, storing them, and then building from the ground up a complete new organism. So reassembling these. So on this picture on the right, you can imagine this would have skin on the top and bottom and a layer of muscle in the middle. This isn't something that exists in nature, but this would be a way to build or design in biology. From a biologist perspective, as a wet lab scientist, this sounds really strange, but from a roboticist perspective, this is really common, right? So people all the time take robots, you can break them apart into scaffolds, actuators, and control systems, and then build an entirely new type of robot or a different robot out of those pieces. So this is really a robotics approach to building biology. This is a very fast video, so I'm going to talk over it quickly, but I just want you, if you're not a wet lab scientist or biologist, to see what this looks like in practice. And so what you're seeing here is these are those frog eggs. These are just fertilized, so they're one cell. And I'm piercing them with a little needle and delivering some RNA. So if I need to, I can change their gene expression. This is 24 hours later. I have to remove a very small outer membrane that protects the embryos. These are normally in water. And that lets me access the stem cells from the top of the embryo, which you're seeing me collect here. They have an outer layer that's very dense, so I peel that off manually, and then with a chemical treatment, I can clean up these white stem cells. I can then pull them into different piles, and then in a well, build them back up from the ground up. So this is sort of like assembling a sandwich, where different layers are different materials or different types of tissues. Another media swap, and you can get all of these cells to readhere into a sphere. And then with very fine tools, a microcautery device and some hand polish forceps, I can subtract tissue and sculpt away to reveal a final design that matches what the computer simulator has produced. And if you're very patient and you do this very carefully, this is what comes out the other end. This is a novel form of living robot. So this is a living robot that was designed by a computer and then built from living cells. This was originally on the left here, the design that I just taken a screenshot of from Sam's work. And this was the first approximation I had built in the lab. This took only 5 days from getting this blueprint to having an actualized design. So these are about 0.5 millimeter in size, quite small. But this approach is really interesting because these morphologies that you imprint are stable, and these legs or these features persist for 10 days. And these live in just regular fresh water, so they don't need any special media, and they also don't need any additional food sources. They're completely self-powered. So another benefit of amphibian embryos is all of the cells in the embryo are preloaded with yolk platelets from the maternal source. And so these have an energy source, sort of like a chicken egg, that they consume as they develop, and this allows them to metabolize that and survive without any additional food in the water. It's not just these quadrupeds. These are just examples of different designs that the AI had spit out. And these are rough approximations that I had built in the lab 1 week later. So we can produce all sorts of different morphologies. And we can even go beyond the shape. We can also introduce different tissues that allow really accurate tissue placement to match the design. So as Sam had said, in his designs, these contractile cells match muscle cells, and these green cells are passive, and this is staining showing where I've placed passive cells and contractile cells as well. And so that's really interesting. We can build these, but do they actually perform in the way that the AI predicts or the simulator predicts? And so we had started by coming up with this pipeline where the goal was simply to move forward, and we supplied the AI with these building blocks. We pulled a number of the best designs and built them and looked at the output. So does this work? These are the first designs that I built in the lab using cardiac muscle to drive linear locomotion. So this was the actual first design I created. And this is just to show that we can layer these cells. The fluorescent red here is cardiac tissue, and that's inside of this passive tissue. The refined design is here in the middle. This movie, if it's playing, there's some beating that's going on, and we've also added some more features of these limbs. And the final design that we produced is this mobile biped that does move in a linear direction, which was incredibly exciting. So this is transfer from the simulated world to the real world. This was all qualitative. We did do some quantitative measures as well. So if you put these designs in the dish, you can track them over time. This is a time-lapse video, and get these motion tracks. And so you can see here this linear motion. So that's represented by one of these lines in this graph. And all that these lines are just different individuals that we filmed over time. And so you can see that all of them move left to right in the predicted fashion. And these pink and fuchsia lines are the behavior of the simulated designs. And so again, here we see great transference that both of these move in the predicted direction. And you can also do a control where you flip these designs on their back and you don't get any linear motion in one direction or the other. You can see that in the video that there's one that's upside down and it doesn't move. And this was really the focus of our first study, is showing this sim to real transfer and bridging the reality gap. I just want to zoom out, and so since we don't have a whole lot of time, we can dig down to this in the question and answer, but just give you maybe 3 examples of the types of things we're interested in from the biology perspective before wrapping up. So just as far as applications, one thing we've looked at is just different types of movements, and can we record experience from moving around the environment. And so we've challenged these designs with different types of mazes, and so you can see here this is a very open maze on the left that's filled with water. In the center, we have an infinity maze where it's more narrow and you have these 3 different wings. And on the right, we've gone down to very narrow glass capillaries. So this is a glass tube that has a 0.6 millimeter inner diameter. So it's about 0.5 millimeter in size. And this is an environment that would be extremely challenging for a traditional robot or even a biohybrid to move through. But the designs we've produced through this design process can move through very easily. We've also engineered a number of memory constructs that we've been iterating on that allows these designs to record information about their environment. So as proof of principle, we started with just simply light. So you can illuminate these different wings of this maze with different colored light. So here we have blue light. And if this design moves through, it actually turns on a fluorescent protein and it starts glowing red, and it keeps that signal throughout the rest of its life. So you could allow this computer designed organism to explore for a set period of time and not even watch it, retrieve it later, you would have a record of his experience. So you can imagine from an environmental perspective, this would be really great. If you could design these to sense pollutants or toxins in a waterway, you could release these at the top of a freshwater stream. You could collect them at the bottom and potentially have a record of the different pollutants that they encountered along the way to do environmental sensing. A second piece that we're actively investigating is self-repair. So traditional robots and biohybrids are typically challenged by injury. They lose function if there's mechanical damage. But what's great about a fully biological system is that self-repair is really a built-in feature of the biology. So on the right here, you can see I'm giving these designs a number of lacerations that are mechanically induced by just simply pinching them with forceps. And these are sped up videos, but within 10 minutes, this tissue heals back completely and you can't see the wound, and you also regain motion and function after that time. And so again, this is not anything that we've programmed. This is a built-in feature of different types of cells from the frog doing what they would typically do, but this gives us a great resistance to wear and tear and damage over time. And part of what we're actively investigating is trying to understand the biology that underlies this principle, and taking these signaling dynamics that happen during repair and porting them back into the simulator so that we have additional features when designing these in the future. And last, we're looking at using swarms to do work. So as Sam had said, there's a number of environmental problems that these may be able to help with. One of the things that we've been looking at is a collection of small particles like microplastics. So there's a growing problem of very small plastic particles that are 5 millimeters and smaller, contaminating waterways and bioaccumulating in fresh and saltwater species. And these aren't captured by traditional filters. But based on our simulation, there was movement types that could be generated that allowed the spontaneous aggregation of these particles. So this isn't a programmed sensor system where these are sensing these particles and actively or intelligently moving them. This is simply a byproduct of the type of motion that we have. And we ported this also into the real world, and you can see here that we have very small particles that are about 10 microns in size, so incredibly small. And based on the type of motion that we can generate, we can get the same sort of behavior. And if you let this run over the course of minutes or hours with all different types of particles, you can get this really interesting spontaneous aggregation or accumulation of particles. And so we're looking at how we could scale this up and how we might even get these designs to capture and hold these particles as they move along as a potential way to do some sort of environmental remediation. So those are just applications. I just wanted to summarize sort of where we're at with the minute we have left, and then just zoom out all the way. And that really, from my point of view as a wet lab scientist, these are the first fully biological robots. And these are designed by a computer. So these are organisms that are no longer under the purview of natural selection and have not been created in nature. These are created in a virtual world and then actualized in the real world. So these probably could not or would not exist in nature, but we can build them nonetheless to solve human problems. They're completely biodegradable, and they're made from amphibian stem cells. They're self-powered, and they can survive all the way from the refrigerator at 4 degrees Celsius up to 24 degrees Celsius in fresh and brackish water. And we've shown they can move, and they can repair, and they can do things like particle aggregation. And just to end, I just want to say that from the biologist and the scientist perspective, I really think that these reconfigurable organisms and this approach is a sandbox technology that can inform a lot of aspects of science. And so from developmental biology, obviously this is a really great system to study developmental plasticity. So we always want to understand how cells communicate, how they self-assemble, and how we can control what they become. And this is really a critical problem in regenerative medicine, is how do we get cells to become a hand or an arm if it's been lost, and how do we get them to grow to the appropriate size and the appropriate shape? In ecology and evolution, there's still a big question about how did we get from unicellular to multicellular organisms, and what are the limits of facultative versus obligate systems? And so this is a system where we can design very carefully and start to tease apart these questions. From cellular computation, there's a lot of interest in how much information processing can you do without a brain or without a nervous system. And so synthetic organisms of all types allow for very precise cell numbers and cell types to be placed. And you can start to ask these questions of, can you build logic gates? Can you build and/or gates? And can you do math inside of a cell? And how many cells do you need? And how much power do you have when doing that? And last, I personally find most interesting is this idea of, in computer science, of how much control should we allow AI have in both evolution and the design process? So we're getting very good at building things in the lab. We can now build parts of hearts, at least, for use in transplantation. And so there's a question of, could an AI design a heart that's better for a particular patient than nature did? So based on the patient's diet, their weight, their lifestyle, perhaps there's a more optimal design that AI could produce. And what level do we want AI to start to get into this biological design process? And I think that's a really philosophical question that I'm happy to talk about in the question and answer. So just to finish, I also wanted to specifically thank two people, Michael Levin, who's the director of the Allen Discovery Center, who really pushed a lot of this research. And that came through also the DARPA L2M program. So this is the Lifetime Learning Machine program, and they funded most of this work. And they've been quite instrumental in providing us support and really getting behind this approach, which was very out of the box at the time that we started. So thank you. I'm happy to take questions.
Okay, great. Thank you so much to both of you for the talk. Before we move to questions, I'm just going to reintroduce you and reintroduce the talk series. We had a bit of an audio issue in the beginning. So again, we are here at Crossroads, which is our monthly event put on by Cross Labs. We are here with Doug Blackiston and Sam Kriegman. So Doug is a senior scientist in the Allen Discovery at Tufts University, and Sam is a postdoctoral fellow at the Wyss Institute at Harvard and also at the Allen Discovery Center at Tufts University. This event, this monthly event series, again, is where we talk about things like intelligence, and including embodied intelligence, like the biological kind of things we've been talking about today, design, engineering, and the mechanisms and mathematics that we might use to understand the sort of intelligence that exists in the world. Cross Labs, which is the institute that I'm part of, is a research institute in Kyoto and Tokyo that's aimed at understanding the mind through computational research. And we explore ideas about brain science, information sciences, artificial intelligence, and artificial life. So stay tuned with us every month for more of this very interdisciplinary talk about, or interdisciplinary talks about the mind, intelligence, and things like that. So with that, I think we'll go ahead and go over to some questions. So for those of you in the audience, the link's over in YouTube for Slido, which is where you can ask questions that we'll then go ahead and ask to Sam and Doug. But before we get started with the Slido questions, I actually have a couple of my own. So one for Sam first. So we've had a lot of talks about evolution and evolutionary algorithms in our talk series. And one of the themes we've discussed is that evolutionary algorithms can often do things that surprise us. So have you had any results that came out of your experiments that you found to be surprising or unexpected?
Well, yeah. So we showed you the final results that worked, but it wasn't smooth sailing from the first initial attempt to something that completely worked when Doug built it out of living tissue. So the first thing we did was we knew we wanted a system that was going to move, and we were interested in these walking systems or crawling systems. So what do you need for a system to move? You just need muscle cells, right? So we built ones out of muscle cells, and we let the AI coordinate their contractions together. And the AI spit out galloping simulated robots across the screen. And Doug looked at these systems and said, there's no way, we cannot build these. The heart muscle is not strong enough to do that. The environment is just not conducive to that. So we had to iterate back and constrain the AI in a way that was more aligned with our goals and the kinds of systems that we could actually build. So that process was manual, and it's something we're looking to automate. And this is a huge open problem in AI, aligning AI with human desires and goals.
Okay, cool. And then for Doug, so you showed us this video of like hand putting together all of these cells. Do you think in the future it would be possible to have some sort of automated process where machines put these cells together? Or do you think that just due to the limitations of the technology that it's always going to be like a human doing that?
That's one of the major goals that we're pursuing right now. We're sending out some funding proposals for exactly that. So there are 3D bioprinters that use different types of inks to deposit cells and build. This is really common in bioengineering. The issue is these are pretty small. So 0.5 millimeter in size is beyond the resolution of most of these systems. And that's not because the technology doesn't exist. It's just because you typically don't want to build something that small in a bioengineering lab. And so there will have to be a lot of R&D, a lot of money up front, that for right now, we're sort of the only game in town that's building these. And so I think we'll do the proof of principle, but these are certainly something that could be built. And if we ever want to scale up to do actual environmental testing, right? I'm not going to be in the lab building 100,000 of these for deployment into an ocean, but a computer could easily build a bunch of these over time. The cells are quite good. So we've played with all sorts of different cells in the lab. I showed frog cells, but you can build these out of other cell types, mammalian cells, and hopefully at some time Piscean cells for use in saltwater. And so that's the other thing we're really working on, is besides just the computer part, the storage part. So can we store the cells for long times so that we have these sort of containers of Lego blocks, and then you could just come back? Because right now also the cells that I'm using are sort of available all the time, but I'm not doing long-term storage, which you would need to do if you wanted to accumulate a huge amount to build thousands of these types of designs.
Okay, great. So then let's jump over to Slido and take a couple of questions. So the first question that we have is from Antoine, and it's a question for Sam. So Sam, the network may generate unrealistic structures such as isolated cells and sparse structures. How does the algorithm get rid of these?
Yeah, so that's some more human intuition that's pumped into the system. So it's not completely an end-to-end AI from scratch. We know that we want the system to produce a single body for Doug to make in this system, although that could be changed in future iterations. So at the end of that network, there's a filter placed on the output that just takes the largest connected component of cells. So you might get a network that outputs 1,000 disjoint bodies, but it's just going to result in one small body, the largest one that it produced.
Okay, cool. Thank you. Oops. So the next question is from Carlos. Have you been accused of playing God, Frankenstein-like aspirations, unethical science, or anything like that?
Doug, you want to take that one?
That sounds for me. No, this is great. This is a great question. There's sort of a lot wrapped up in this with the ethics and the design aspect. So certainly you hear a bunch of things because while this invokes sort of a lot of creativity, this also has sort of the Terminator vibe and all these science fiction movies, right? This is the start. You build this thing that the computer designed and it goes wild. From a technology standpoint, though, this is incredibly benign. So number one, you can think about this. If you've ever had a skin biopsy taken or know somebody that has, there's more cell types and more complexity in there than in any of these xenobots, which are just skin cells and heart cells. There's no neurons. And so if you're not worried about your skin biopsy or skin biopsies taking over the world, these are pretty mundane. The second part is that these are, from the playing God, these are not genetically modified. These are just cells that exist in a dish. And so compared to even what people do in a brewery where you are genetically modifying yeast, or you are in a lab and you are genetically modifying bacteria, I think there's the regulation and the dangers of those types of systems have been heavily worked out over the past 40 and 50 years. So we know exactly what we can and can't do. And certainly from our point of view, as scientists, I can also say that all work that we do with biological cells or any biological material has to be approved at the university, the state, and the federal level. So there's oversight, and that has to happen before you begin any of the studies. And so we have to ask for approval, and we have to make sure that it's safe for humans and that it's humane for the animals. And that also acts as sort of a definite extra set of eyes at multiple stages to just do a sanity check. So yes, you hear these things and I think they're fun, but in the grand scheme of what people are building with synthetic biology, these are actually a pretty basic system when you actually come down to it. It's some skin cells and some heart cells that have been assembled in a particular way.
Okay, great. Thanks. So our next question is from Olaf. How much can xenobots adapt and learn so far in your best instances, and how smart can their decision-making be with this method?
Doug, you want to take that one? Just talk about
Sure. Maybe you could talk about on the AI side also what we could get them to do. On the biology side, I will say that there are opinions differ across the biology world about how much these might be thinking or processing. So they certainly have some ability to sense their environment. This is not programmed. This is a built-in feature of the biology. As far as adaptation and sort of intrinsic memory and learning, if you were trying to talk about consciousness, I would say we don't know. Right now, everything that we've seen is largely just physics-based that we can predict, but they certainly do have the ability to receive inputs. So we've shown that when you injure them repeatedly, there are signals that change over time. So they respond differently to the second injury than they do to the first. And so whether that's some form of learning or adaptation, we don't know, but we can detect differences in signal processing with repeated events. We're also looking at things like stressing them with salt concentrations or things in their environment. And metabolically, can they turn on pathways that would help them respond to these different environments? As far as having goals and sort of hopes and dreams, I would say presently there's no evidence for that. That doesn't necessarily mean that that doesn't exist. But what we've seen is largely a byproduct of the predicted types of motions that we would see, which also is borne out by the simulator, which is it does not have adaptation built into the final design. It's simply running off of physics.
Yeah, so on the design side right now, I'm a roboticist by training. So I think of these things as open loop control robots. They have motors, but whether or not they have sensors and whether or not those are connected to the sensors, to the motors, in simulation at least, that doesn't affect their behavior. So we could add closed loop control in which the sensors affect the motors, which affect future sensory repercussions, and that loop goes back and forth, and you can get some more interesting targeted behavior. That's something we're investigating now. But you might just view these as one-off kind of custom robots for a single task that kind of are blind for the moment. So are those intelligent, or how much are they intelligent if they can not really sense at all and only act in a very limited way? I'm not so sure, but you could also view the AI system, this whole pipeline, as some kind of intelligence. It has the ability to output all kinds of different creatures that do different kinds of things. So it can act in the world by creating a very specific kind of entity. And it can do that in a diversity of ways. And depending on its sensory apparatus, it could change the way that it is creating these systems and make hypotheses about what would be better systems. And that sounds like a very intelligent system to me.
Mm-hmm. And I also add that we're definitely an area of study is designing these to have some sort of behavior that can be programmed. And so from the biologist perspective, you could imagine, say, if you sense this chemical, swim towards it or walk towards it. When you get there, produce a novel compound to decontaminate it. At the end of your life, swim to the surface and be collected so that you have a record. Like that type of logic exists on the biology end already for bacteria and yeast. And we could port it into the system, but it's not trivial. It's going to take a little bit of time.
Well, it definitely sounds like you have some fun research directions up in mind. So that is really cool. Okay, our next question, oops, sorry, I accidentally marked that one as answered, but our question for Madi was that the precision of the technology and the ability to put the cells together in the right way is basically dependent right now on the precision of the technology, right? Like on the biological level.
Yes. So right now, we've done some statistics to look at how well we can match the designs, and I believe roughly you could imagine within 10% error in either direction, we can match the placement of the tissues. That's another part that we're actively investigating as well. Is there's a lot of different ways to micro pattern, so you can make very small sheets that have textures or chemicals on them that make certain cells stick, and that would give you greater precision. The precision right now has not been limiting to us, so we actually find that our models are abstract enough that even with a small amount of error, you still get good behavior. You don't have to have exact one-to-one match. And currently, we've been much more interested in speed rather than precision. So we want to create a lot of designs very quickly to screen because we're at the early stages of the project. I'm certain that as this progresses, we will be more interested in patterning finer structures or getting better resolution, which will also allow us to increase speed of production. And that's something that we'll be moving into probably in the next year or so.
Okay, cool. So here we have another question for Doug from Antoine. How do these AI-generated biological robots compare with the ones that are manually designed, say, in terms of speed of design and build efficiency and things like that?
That was probably one of the most shocking aspects of the project, is that this worked so well. And so the biological designs, in my opinion, at least for the first paper, performed better than the artificial constructs. And we think that's because of something that Sam already alluded to, is that biology does have some levels of self-organization that the AI did not have included. And so cardiac muscle tends to contract in these contractile waves, and those pulses allow the generation of more efficient movement rather than individual voxels turning on and off. And so some of that we are learning how to port back into the simulator. Overall, the AI is fantastically better at me than design. So it takes me about a week to a week and a half to build these, and I can build 20 to 50 at a time. And that is for one design. And so the AI in that time has gone through tens or hundreds of thousands of iterations. And so it would take me a lifetime to even go through one experiment that's happened in the virtual world. And so what's wonderful for me is that while I'm taking orders for a computer, which seems a bit strange as a biologist, the speed that we've been able to make progress on this project would not have been possible without the AI. I would still be building my original versions. I would have no idea what to build, where to place the tissue. It would be a very blind way to go into the research. So I think what's been interesting is the transference has been so strong, it's allowed us to make progress very quickly, that the predictions have generally been borne out in the biology from what we've seen in the simulator.
Okay, thank you. Next question is from Bert. So the swarm movements look random for now. Is it possible to evolve desirable swarm behaviors using genetic algorithms or other methods?
So we have shown in, I believe, the second paper, which was in Science Robotics earlier this year, that you can, it's difficult sometimes to predict individual behavior. We have different versions of these xenobots, and they move with different kinds of motors that we didn't talk about here. They can also use cilia, which are like little hairs that allow them to swim instead of cardiac muscle, which pushes off the ground. It's difficult to predict the swimming behavior of these things, much more difficult than the cardiac ones, which is why for the first paper we used cardiac and deciliated skin tissue. But that said, we can predict some interactions with the environment with a swarm of individually unpredictable xenobots. So we were able to show that although the movement of the individuals was unpredictable, the way that they pile synthetic particles in their environment, the size of those piles, where those piles are, how efficient they are at doing this task, was predictable based on the shape of the individuals in that swarm, which was interesting for us. So this turns the problem into a kind of statistical like mechanics or probabilistic situation where, depending on the shape of the individuals, and they're bumping into each other in kind of random way, but they have interactions that tend to happen with a certain shape more than another shape, and you end up getting something on average that could be useful, like a large pile.
We're definitely looking, so in the biology side, we're actively looking at that. That's part of a paper that will be in review soon. But we can build different shapes that have different piling behavior. So some shapes are more effective at creating piles, some are better at not making piles at all. And so you could imagine in the future even having combinations of different shapes that have different abilities. And you can have this sort of additive math where group one makes the pile, group two collects it and picks it up and does something else with it. So you could get this really interesting additive behavior simply by changing the shape and structure rather than the individual behavior or movement types of the individual xenobots.
Right, cool, thanks. Next question is from Carlos. How feasible or useful would it be to build macroscopic xenobots or biobots?
We started exploring this a little bit in a conference paper that will be presented next week at Robotics, Science and Systems Conference, which is held virtually. We explored what we were calling fractal robots, which is this idea that you can have robots inside of robots and they can behave similarly at the different scales. So Doug was demonstrating this, and I'll let him explain out of these frog cells that you could have spheres of different sizes and connect them together in different ways to produce similar behavior at different size scales, and potentially much bigger than just one of these bots alone.
Yeah, so the two design principles that I've tried is you can certainly make them bigger just by adding more cells. And we've gone up to 1 millimeter to 2 millimeters, but there's no reason that you couldn't go larger. The issue is that starts to become biologically expensive as far as the amount of material you need. And there's some odd things about these are pretty soft tissue. And so if you want to make a structure like a sphere or cube, at a certain point, you just end up collapsing under your own weight. And so you can't hold the shape as well. So the other idea that we pursued is this fractal idea is, could you have individuals that connect and sort of build like Lincoln logs into larger and larger structures? And so these, up to a certain point, are quite sticky. And so if you give them a small injury and stick two together, they will stick. And you can essentially daisy chain these together to make larger and larger structures. And then you can multiplex those and build cubes out of them. And so that's probably a better way to do this. And you could even imagine at some point, this is still science fiction, but the technology exists that you could program them to do that. So you feed individuals through a small tube. You have them connect into a large super robot. And at the end of their lifespan, they break down into individual components and swim away again. I think that would be a really great application of the technology down the line.
Yeah, that definitely sounds really cool. Have you tried injecting xenobots into a live xenopus frog, and did the modified cells maintain their structure or change in any way?
So you can, we've tried this with a couple different cell types, and it works. You have to stay within organism, obviously, because of the immune system. Frogs don't go directly from egg to frog. You have to remember there's a tadpole stage in between. And the tadpoles take very long to grow up in our species, about 9 months. So we typically work with the tadpoles. Tadpoles do not have an adaptive immune system. So they only have an adaptive immune system after going through metamorphosis. But if you do place a xenobot inside of a tadpole, it survives just fine. If you place a xenobot into a developing embryo, you can actually get it to become part of the embryo. It heals shut and then becomes part of the skin of that organism. But otherwise, they do persist. Frog are, I wouldn't say that that's necessarily surprising. So out of all the species, frog is incredibly amenable to what we would call cut and paste experiments, where people have known before too, you can also place beads in the skin, which give off chemical signals that help stem cell differentiation. But you can absolutely implant these. They are too large by far to fit into blood vessels. So even if you were to think of making these in humans, one cell of a xenobot is about the size of a blood cell. So you would immediately have blood clots if you tried to use these intravenously. But in sort of the pericardial cavity or just in sort of the integument of the stomach, these can go in and they survive just fine.
Right, very cool. That is sort of related to another question we had about whether or not it would be possible, for instance, to use xenobots in lungs, like for cleaning, removing pollution, things like that.
Absolutely. So there's someone else in our lab who's trying to do these using different types of mammalian cells. I can't speak to her research specifically because it's not published, but she's using different cells and exposing them to different microenvironments and culture, so organoid systems and so forth, to show proof of principle that these would work in human medicine moving forward. And so far, all signs point to yes. Hopefully that paper will be out this year. They're just finishing it up right now. But certainly, you could imagine using these in a biological system. The trick is really the programming part, which is sort of what we've come back to a couple times, is programming them to go to a specific location or respond to a specific type of injury. So we've talked about things like introducing them to the site of a spinal cord injury, having them seek out sites of damage and release neotropic factors to help with nerve regeneration. And then because these are built out of mammalian cells, they either differentiate at the end into nerves themselves, or you allow them to be cleared by the host immune system. Technologically, that's possible, but again, the details are often very challenging to work out on the biology end.
Makes sense. Yeah, we had a lot of people very interested in basically how compatible these xenobots would be to work in the human body. I think we've covered that at this point, so we'll skip that question. The last question we have right now on Slido, which again, if anyone has any last minute questions they want to ask, now is your chance, so go ahead and drop those in Slido. Going back to the sort of swarm intelligence things, are there certain categories of xenobots at the moment with certain behavior for swarm intelligence studies, or do you just have sort of one kind of xenobot that you're using for that?
Well, yeah, I'll let Doug describe the details, but I was kind of trying to very quickly explain a whole other section of xenobots that we've been exploring. So there are the walking ones that use heart muscle, the swimming ones that use cilia, and we can add all kinds of other motors and sensors into these things. And they could be used in a swarm together. Depending on the motors they have, they're going to be faster, slower, interact in different ways. And this is an area of future research for sure.
Yes, so I will say on the biology end right now, one thing we're trying to do is engineer specific types of motion. So right now, the type of motion from these swimming xenobots is fairly random per individual, but predictable within an individual. So we have individuals that move in a straight line, individuals that circle, individuals that take arcs and do these interesting behaviors. But the ones that do those behaviors keep doing them. They don't change behaviors all that often. So the method right now has been sort of to survey all of the bots that are generated and pluck the ones that do the behaviors that look interesting, and we can use them in the swarm, or we just pick randomly. On the flip side, what we're trying to do is, rather than do that brute force method, is to specifically engineer those types of behaviors. And that's proven to be fairly challenging. There's a lot of self-organization that you're fighting against in the biology, where there are cells that are given signals to align in certain directions or point in certain ways. We have the tools to adjust those, but it takes a lot of effort at the scale because these cells are in a very odd configuration. So they're trying to interpret signals that aren't necessarily correct, which is sort of driving the patterning cues in odd directions. And so hopefully in the future, we'll be able to specifically pattern the angles of these cilia and generate specific types of motion, or even be able to control the motion. So based on lights or signals, they can change the type of motion that they have. But right now, it's just simply by selection, sort of either handpicking or using random motion.
Great. Next question. So what is the first thing that can be done with xenobots for society or the industry, say, tomorrow?
I think maybe the idea of computer designed or AI generated biology and technology might be the most applicable idea right now. I'm not sure when xenobots themselves or some kind of progeny of xenobots made out of other cells with other kinds of things would actually be useful in the real world. It's very difficult to predict those kinds of things, if they're ever will be useful. But this idea of allowing computers and AI to design functional physical technologies in the real world, I think is really powerful. Just as one example, planes have pretty much looked the same since they were invented by the Wright brothers. I mean, okay, maybe they're slightly different, but there's two wings, you know, there's the same basic shape. But I'm sure that there are infinitely many designs that can fly, and probably much more efficiently than the planes we have right now. And there are government agencies that are interested in this kind of research, and it's funded. And I think that this paradigm of AI designed objects is going to start taking more ground away from humans, but not necessarily just AI versus just humans. I mean, you can work together, and they can be a really good combination together with an AI helping a human be a little bit more creative and try out designs that they wouldn't have tried out otherwise.
Yeah, and I would say from the biology end, the two things that we've talked about are still pretty far off. So human testing is quite far off. To get to clinical testing from a money and regulatory standpoint would be more than 10 years off at least. And so you would start in mouse and work your way up, and there's a lot of regulation and careful planning you have to go through. Environmental testing is a little bit easier, but that still requires, so right now we cannot take anything out of the lab. We would never test anything in waterways. That would require working closely with one of the government organizations, the EPA or the USDA, to start this sort of a pipeline to do environmental sensing, which I think is something that could be useful in the future. So either sensing pollutants, or even if you wanted some rare earth metal to build cell phones, could xenobots soak those up and bioaccumulate them, and then you collect the xenobots and you have a lot, as opposed to sort of trying to pull these trace elements out of the waterways. That would still be 5 to 10 years away for sure. And I think, as Sam said, the bigger thing for me is there's a whole field of bioengineering that's getting really good at building human components in dishes. So cartilage to transplant into people's knees or arthritic joints, and heart valves. And that field is still pretty far removed from computer science and this idea of AI. And I've seen it almost never. And I really think the language needs to come together. And I think there's a lot of innovation that the computer can solve that, again, sort of like if I was trying to design xenobots by hand, this brute force method is very slow. I think that we can make a lot of progress on the bioengineering end if they incorporated more of the computer science and AI into it.
Yeah, as I've said many times, because it just continues to be true, there are so many cool future directions for this kind of work. That's really great. As one final question that we'll take here off of Slido, do you imagine that xenobots could actually be made smaller so that you could, for instance, put them on skin and have them be absorbed into the body?
You can make them as small as one cell. That's currently the smallest you can make. There are a number of groups that are making smaller things completely synthetically. So basically making artificial cells from scratch. There's a lot of work at the Wyss Institute looking at completely synthetic cells built, the DNA and everything from the ground up. Right now, they wouldn't be able to go through the skin. Your skin's pretty good about keeping microbes out. That's a built-in feature that you have that's keeping you from getting contaminated from bacteria, which are much smaller than frog and human cells, and viruses and so forth. We could get them down to probably, a single or a small group of cells would be 20 to 30 microns, which is quite tiny. That's still, we're still talking 10 times bigger than a red blood cell, though. So these are still quite large from the point of view of microscopic things that you would be using in a human. These aren't nanoparticle size. These are bigger. But on the flip side, I would say the advantage there is that we get a lot of biochemistry from those cells. We can do a lot of different features with different cells, and we can engineer a lot of computation by having a multicellular construct as opposed to a single cell. So as of right now, the short answer is no. I don't think they'll be able to go through the skin without some sort of help through a needle or some other process.
Okay, great. Thanks so much, and thanks to everybody who asked questions and who joined us today. Before we jump over to Zoom, which, by the way, the link is in the YouTube chat, I just want to go ahead and give one more thank you to our parent company, Cross Compass, which is a leading AI company in Japan working in technologies such as manufacturing and other areas of artificial intelligence applications.